And now you can only try to match that typing with the idea of going gently. The next speaker is Zam, otherwise known as Zam Iqbal, talking about species identification.
Hi everybody. Right, I'm Zam. I should say from the start I'm very happy to have questions all the way through. So this is my second time at a Newton Institute program, where Wally was at last time as well. And the thing that struck me most last time was that I liked the fact that people were giving talks about stuff that they were thinking about rather than stuff that they'd done and they wanted to show off. And so when I had to write an abstract, I thought about something that I wanted to be able to do rather than something that I could do, assuming that I would have sorted at least some of it out before I gave a talk, and that's not entirely true. But what I'm going to do is I'm going to talk to you about what the problem is, what the naive thing to do is, and why I thought the naive thing wouldn't work, and what I thought you ought to do, and then why, annoyingly, the naive thing seems to work, essentially. So, okay, so what's the problem? So Staphylococcus is a type of bacteria. I'll talk about that in a second. And the problem is, can you identify the species of a Staphylococcus once you know that the thing that you've got in front of you in the culture plate is some kind of Staphylococcus? And there are medical reasons for doing this. The different species have, well, there's one particular species called Staphylococcus aureus that's much more well known for being pathogenic, or sometimes being pathogenic. And there's a whole host of other ones that are often commensal with us. And when a doctor has a patient who's ill and they have a sample from them, you know, one of the things you have to do is determine what you've got and then how you treat it. So, okay, we've got so you've got some data from medical source. I want to identify the species type. But the problem that worried me, and the thing I wanted to talk to you all about after the talk was the fact that there's these strong biases in what we know about. So there's basically, I don't know how many, Nick will know, that more than 50 known reference genomes of Staphylococcus aureus, and there's about one each of a whole host of other ones, so 40 or so. So I wanted to know how I was going to handle that, along with the fact that there's this thing, the accessory genome. So this picture is a guy called Phelim Bradley, who did a lot of the work in this. He's a PhD student in my lab. So what's my intuition when I went into this problem? I thought, okay, you've got genome sequence from some kind of species, and I know that it's easy to compare genomes of species looking at assembly graphs. There are different ways of doing it. I have one, but there are several ways of doing it. So it ought to be possible up front to determine species informative k-mers or contigs, something like that, and then just look at your data and wait till you see things that you recognize. So I'd be happier if I was giving this talk after having heard Daniel's, but anyway. You may have heard of the Kraken software that came out earlier this year that has a very interesting idea where basically you just map k-mers to the lowest common ancestor in the taxonomy of where they exist. And so when you get reads one by one, if you have a k-mer that's informative of k-mer, sorry, it's a word. If you have a DNA word that sort of is informative of your species, you can remember that. So that sounds very sensible, but what bothers me is the fact that you only have one reference genome for lots of these species, and we know there's horizontal transfer between these species. We know there's sort of big elements that move backwards and forwards. And so what worried me was that you'd look at things, and this thing would look like it's in, this belongs to Staph hemolyticus, because it does, it's in that reference genome, but actually it's polymorphic in that species, and it's not informative, and maybe even worse, it's in that species, but it's actually in several of them. And so I wanted to know how much confounding that would cause if I went and did the stupid thing. Okay, so just a quick thing about what are staph and why should I care. So they're gram-positive bacteria. They live on the skin of mammals and birds. And so Staph aureus has sort of two lifestyles. One is sort of very easygoing. They're asymptomatic. So 30 to 50% of us apparently carry them in our nose. But then there's an acute form where it actually can colonize a whole host of different tissues, so you can get a whole host of different illnesses depending on where it's ended up. There's another form, I'm only going to mention a couple of them, but Staph epidermis lives on your skin. So at times both of them can live on your skin, and it's known that there's horizontal transfer between them, and there have been. And it's fine when it's on your skin, but unfortunately we do things like we operate on people, or we put catheters in people, or we insert sort of new joints in people. And if you do that in the process, you sort of introduce some staph epidermidis into you, then it's pathogenic. And in fact, at least one reference I've managed to find claims that there are more nosocomial meaning in a hospital, bloodstream infections are caused to staph epidermidis than Staph aureus. Staph aureus is the thing where if it has resistance to methicillin, it's MRSA, which is the superbug that you often hear about in the news. So here are some pictures. Staphylococcus coming from the Greek for looking like grapes, essentially. And these are, that's Staph aureus, that's Staph aureus. No, that's Staph aureus being eaten by a leukocyte. Saprophyticus epidermidis, I think. Doesn't matter. So there we go. First known staph infection. Plague of boils, 6th plague of Egypt. Obviously I don't really know, but I thought it was a nice picture. So historically, how have we typed them? So Koch identified them in 1878, and he thought that they correlated with abscesses. And not long after, Ogden showed that if you took pus from a human who had an abscess and stuck it inside a mouse, then the mouse ended up getting an abscess too. And from then until the early 20th century, essentially you differentiated them by colony colouring, which meant that actually we were lumping a lot of different species together just because of their colour. And then basically you start accumulating a host of biochemical tests. You can either look at whether or not they have a coagulase, you can see whether or not, you can look at the different properties of the cell wall and so on, and essentially these are all things I know nothing about. But I know that there are a host of different ways in which you can essentially develop a whole list of classifying properties that you think a particular Staph aureus, for example, or a Staph epidermidis ought to have. And then into the sequencing or the DNA era, there are ways of looking either at the ribosomal RNA or MLST or whatever. And if you look at the actual genomes and you wonder how different they are, then these guys are, so that's that little bar there's 5%. You just have these guys are Staph aureus, and these are a few other guys. I don't really want you to take anything concrete from that. And the thing that worried me, as I said, is that although 80% of the genome is preserved between these species, and synteny seems to be preserved, I haven't checked that very carefully, but it seems to be, there are a lot of different things which move around either within species or between species. And if you start digging around, you can find examples of things that move between species. So, okay, so what did I do? The first thing I tried was just imitate the microbiologists. So the standard thing, if you read a medical textbook, is if you're trying to classify a staphylococcus, you do a catalase test and you do a coagulase test and see if it's positive for both of them. So catalase and coagulase are, these things are produced by genes. I ought to be able to just do a gene presence test for those two. So I look for the genes that create those two things. And then all I do is, you could do this any number of ways. You could map your reads, or you could do an assembly. I just did it by assembly. And then I just take a whole load of samples and see if they have if they're cat positive and coagulase positive, then it's Staph aureus. If they're cat positive and coagulase negative, then they're some other staphylococcus. And that's actually often, crudely speaking, that's the first thing that medic cares about. Is it aureus or not? Okay. So I've tried it on, again, this is the problem you have, is you have quite biased data. I tried 501 Staph aureus, and all of them are, and it works, and it tells you the right thing. And on 40 non-aureus reference genomes, again, it's correct. So, okay, that's great. It's not massively impressive, but it's interesting that you can just go and imitate the microbiologist, essentially, on your computer.
Maybe that's what they do. If they did the same thing as you, then... That's not.
I'm really confused.
Don't worry, keep going.
Okay, I'll keep going. All right. So this is the thing. This is sort of the main the thing I want to really talk about. So, supposing I want to do assembly, genome assembly, and look for contigs or chamers that distinguish the species, how am I going to do it? So I'm going to give a description of a method that I use. I don't think the method is particularly important in some ways, but I want you to get the intuition. Can you see that? Wonder if this works. Doesn't matter. So the bottom line is this. I've got two chromosomes, say they're two species up at the top, and I've got in red characters where they differ. And all I want to do is break up the genome into a sequence of words, let the sequence of words create essentially a graph or a network, and look at how those graphs or networks differ between species. So along the top, I get the words that are created by the top genome. Along the bottom, I get the words generated from the second genome, and they create some structure. And if I look for those structures, they're differences between genomes. So if I zoom out, I create this cartoon. Take another example. So these are two different species. That GCT difference between them creates this bubble, but there's a GCT somewhere else in the genome, and so the graph has to come back and touch. So it looks a bit messier. You zoom out, you get this. So if you do this for the whole genome, you get something like this. But the point is I want to do this for lots of species at the same time, so I remember, that species 1, that species 2, and I can just go backwards and forwards. And so the idea, my intuition is very simple. I just look for bits. If I'm interested in the blue species, I look for things that only exist in the blue species, and so on. And fine, you can look at where the chromosomes are. So the problem is that my known references are not uniformly sampled, and I know that there's stuff shared between the species. So what my concern was, if I have an unsupervised method, it will just pull out anything, and some of them will be more informative than others. So the whole of this talk basically was supposed to build up to saying, I want to talk about this to you about this, look sort of in the bar afterwards. So the idea is that I want to allow for polymorphisms, so things that seem to be fixed substitution between the species. And so what I thought of doing was following this paper by Wilson et al., where essentially they want to integrate some knowledge of the phylogeny with your knowledge of population genetics and how you expect the frequency distribution to work. So what they do is they conditional on the ancestral allele and integrating over what your allele frequency distribution is like. They were trying to work out, look for signals for positive selection. And I thought I could somehow integrate something like this. So the plan was, do the stupid thing, show it doesn't work, try this, show it was better. So okay, I should just do it. That's enough talk. Okay, so I create the graph of, I'll take 25 staph species, including 6 staph aureus. I look for regions in the genome I look for bubbles in the genome where one of the alleles is specific to one species, and the other side I don't care. But basically what I'm doing is I'm looking for bits of the graph that uniquely exist in one of those species. And then when I get a sample, I will just see which bits of the graph light up. And on the basis of, just even something very simple, just a majority call, then say what species you've got. So this is the length distribution of the things I find. I mean, you can't see this, but nearly everything is a SNP. But the biggest things go up to about 2.5 kb. And the long things are things I will tend to not trust, because I essentially don't really trust mobile elements. And the SNPs I might trust unless they're SNPs between mobile elements or copies of mobile elements. And so I run on a 500 staph species and it works, that's fine. And then I basically start digging around in the dustbin of the hospital for samples that they threw away because they're bad for some reason, and that's not staph aureus, so we don't care, or that's mixed, I don't trust that. And it sort of seems to work, basically. I basically can't find a problem. It's sort of annoying. So validation's underway. So I started thinking about why isn't it a problem? It ought to be a problem. So this suggests I should have read more papers before I tried. This is a plot showing a sort of reciprocal blast between a whole load of staph species. So all the white ones are staph aureus. These two are staph epidermidis. This is hemolyticus, this is saprophyticus. So the colors, okay, if something is colored white, that means the nearest match to that chunk of genome is something else within its species, or in fact, within its clade within a species in that case. This light grey tells me that the nearest, these two are the same species, and the nearest match to them is within the species. These guys are the sections of the genome where, or the proportions of the genome where your best match is something in another species. And actually, I mean, basically the proportion of the genome, the accessory genome of staph is just not big enough for me to need to worry about this, is the bottom line from the end of this, I think. I think I've been talking quite fast. I've actually almost reached the end of my talk. That's frightening. I should have danced or something. Yes, I go back to the start. Okay, so it seems like the simple methods work, which is annoying. And the thing that the more observant of you will have noticed is that actually no metagenomics in what I've just done. Okay, like all of my samples, essentially if you go in a hospital and you get something from a blood culture, what tends to happen is you grow something up overnight in a hot thing, and you wait until it's generated enough carbon dioxide that you think it's grown enough, and then you pull out and you pick from something that looks like a colony. So you're highly biasing away from a mixture. So unless you're looking at a very fine level, basically my input data is biased against mixtures. On the other hand, when it does happen, so there are situations in staph where what you have is the major population is sort of not drug resistant and the minor population is. And so there are situations where you're very interested in those mixtures, and so that's the kind of thing I'll be working on in the near future. I wish I hadn't given my talk just after Richard. But anyway, that's the end. I've even forgotten to put an acknowledgement slide, but the main people to thank are Phelim, who did lots of this work, and lots of work that I haven't been able to talk about yet, and Derek Cook's lab at the John Radcliffe Hospital in Oxford. Thank you.
We have time for questions.
We certainly do.
I didn't quite understand why you're not interested in insertions or accessory genes, or presumably antibiotic resistance plasmids.
I'm extremely interested in them.
Well, that's what I thought.
I'm extremely interested, but I don't want to use them as a way of telling me what species I'm looking at.
But they're not dependent on species, but they're very important. So does this sort of method pick up the fact that one isolate has multiple copies of a, let's say, methicillin resistance, and another one doesn't?
It is. So that is precisely what another project that I'm doing. So you can, I mean, a nice thing about this method is that you can apply it to both SNPs and to indels and to plasmids and to all kinds of things. So you can look for presence and absence of things like that.
And can you do them at the same time? Because I mean, they're much bigger than a SNP. You're looking at possibly up to 100 kb or something.
You can. Yes. Ask me again in like 3 months, and I'll actually have some results. I'm still in the middle of analysing a whole load of data. But I mean, it does work. But I mean, there's two problems here. One is the genotype-phenotype map. You've actually got to get the association between the thing and the resistance. And actually for Staphylococcus, that's quite well known. But then the other thing is, once you know what you're looking for, can you find it? And so I'm talking more about the latter.
Yeah, so I have I guess more philosophical question. So people have been trying to do this on a regular basis in clinical practice for at least 10 years now that I know of. I mean, it's either biodefense or clinical practice, and no one has declared victory yet. So if it's that simple, how come it hasn't been done?
So so I think there's two questions. It depends on what you're trying to do. So if you look at that the massive biodefense thing that happened last year, yeah, I mean the input data for there was much more challenging than what I'm trying to do, right? That essentially you have to be able to handle any species mixed in with any host, and you had to be able to. Ideally, you wanted to be able to sort of identify stuff which could be there at very low proportions.
Yeah, but if I may, I mean, even in terms of, say, methicillin resistance or something like this, there, as far as I know, I mean, there might be, there is no test that I know that just simply sequences or PCRs out that gene, rather they go to the whole assay for methicillin resistance. So even that exercise of saying, here's the gene I know, doesn't seem to succeeded in clinical practice, at least.
But it has a lot to do with certifying the process, right? There may be
As
legal
well.
reasons that's not maybe.
So there are two things. One is the certification is non-trivial, but also, so the genes that you look for can often be very diverged. So it's not like you can, it's not like you take an example. If you took a human exemplar gene, essentially it'll be almost identical between everyone else in this room. Whereas if you take, the beta-lactamase gene in staph, which determines penicillin resistance, and you do multiple sequence alignment, they can be quite different. So there's a question of what you're looking for, which adds a layer of complication to that.
It's more just that bacteriologists don't want to do molecular-based testing because they like culturing organisms. And that's what they used to. They could certainly do this now if they wanted to. And some groups are doing it, not routinely, but more commonly. But you could do it. It's just
Richard.
they don't.
What's up, Richard?
I heard somebody saying this. So I think for STDs, sexually transmitted diseases, they now have non-culture systems for identifying them. This is a standard technology. Maybe the people in the room know more about this.
Yeah, things like chlamydia.
Yeah,
Because they're much harder to grow.
in the UK. And so people are taking swabs, and there are direct assays, but people are also looking at retrieving DNA in bulk, which is essentially a metagenomic sample, and identifying strains from that, which would fit exactly into this framework. And I think that is the way that things are going to go, including in the clinic.
Yeah. Absolutely.
So, and I think, you say it's all too easy, but that's because you set yourself a challenge right at the top of the tree. There's a lot of very important information much lower down not about plasmids for sure, and also about which strains are present, which is really valuable in a clinical setting to know.
I'm not arguing against that at all.
So that, I would recalibrate your interest into following which particular Staph aureus strain have I got here, or Staph whatever.
But Richard, I'm
What are the
giving a
prospects-
talk
what are the-
here
what are-
about a thing that I've not been focusing on.
Okay, what are the prospects? What are the prospects for doing that? For essentially strain identification.
That's completely doable. I mean, that. I think that's actually doable already by almost any method you try, especially for staph, because it's not. I mean, in terms of determining its strain type, where you're essentially looking at the core genome only and using SNPs, that's completely doable. And I think people already have them. I mean, there are ways you might have for making it faster, if you see what I mean. But so my perception is that's, and I'm not saying that doesn't mean it's worth doing, or even that I'm not interested in doing it. But I think that is a problem that is soluble, or even solved.
But I think an interesting opportunity with the genome sequences for medical practice is the detection of kind of hybrid strains, if you like, where you've got a new mobile genetic element that's seen in a species for the first time, and there's lots of examples of recent emergence of antibiotic resistance determinants like NDM1 and KPC and things like that.
Yeah.
So how feasible is it to put all bacterial sequences ever sequenced into one whacking great graph and be able to detect rapidly that some mobile genetic element or plasmid that's only ever been seen in some particular species is now made its way over to so Klebsiella plasmids now and an Enterococcus for the first time, and do it with your framework? Is that doable? Can we just have a pan-genome of everything in one graph?
So it depends on how big a computer you have, unfortunately. So so I built a graph of NT last year, and I think that had 22 billion k-mers in it or something like that. So I needed, so in terms of RAM, I can't remember how much I used, but it was over 256 gigs of RAM, and it was less than 512.
That's not too. That's
So
that's
yeah, it
that's
depends.
commodity, said Daniel yesterday.
So you can do that. You can do that, and So to be honest, I've been thinking more about working within species than trying to hold everything at the same time. But you're right. So if I do that, then I won't spot, Van something has moved from Enterococcus to Staph or something. These things are doable. It's just a question of how to make it. There are two options, right? Either you just want it to work for your lab, okay? In that case, you can do it, and I can set it up for you. But the other thing is if you want everyone to be able to do it, and that's a lot harder because really, actually, then I think that's too much memory, and you need a data structure that's a bit more efficient than what I've been using, and some things, or some sifting mechanism that tests things one at a time, and that's doable. And that's actually puttable on the cloud, probably. Yeah.
Two questions.
Okay.
First, there are 49 species of Staphylococcus right now. Have you tested all the species within the genus?
So, no, that's next on my list. And actually, we're going to be sequencing more species within the genus, at which point I'll put them all in.
Because the thing is that if you use the identification that you get from a clinical laboratory, there are typically two things that you'll see. You get differences not only within the systems that are used in different clinical laboratories, if somebody's using, say, a biolog system or bioMeru or somebody else, but you're also going to see a lot of strain variation. And that strain variation is oftentimes very specific for a hospital. You see the same sorts of things with mobile genetic elements and antimicrobial sensitivity. Each hospital almost has its own unique patterns. You'll also see, to the best of my knowledge, a certain degree of, say, fuzziness in the identification. Because the strains that come in from the wild are always going to be a little bit different than the strains that were tested within the system that's used for diagnostics. So how do you actually use your test and then prove to the extent possible where you're really confident that your identification method not only works and accurately gets what you want versus something that you think is working but has a little bit of free play in it because you see things that are moving across, say, the species within the genus or maybe even across into different genera that are closely related.
So I think you're effectively asking for two things. One, you want a method that doesn't just give you an answer, but that gives you a confidence. So you want a proper statistical model behind it. And two, really what you want is a method that effectively grows as, basically you don't make a fixed model that you then carry on using for the next 5 years. Every day when you get a new sample, you should be able to add that and use that information to sort of improve future identifications. So that is, so the second thing is, well, actually both of them are precisely what I'm trying to do this year on staff. The question of on the fly updating your opinions about mobile elements moving between species, that's hard. I'm not sure. That's probably in my list for version 2 rather than version 1. I'd be very interested to talk to you about that afterwards, actually.
Any more questions? No, thank you.